بسم الله الرحمن الرحيم والحمد لله رب العالمين والصلاة والسلام على أشرف المرسلين سيدنا محمد عليه أفضل الصلاة وأزكى التسليم. Dear students, today we have a lecture and the title of our lecture today is Feeding Behavior and its Disorders. After we finish the gastric secretion and the gastric motility, now we are going to another topic, which is the feeding behavior and its disorders. If we look to the feeding behavior is considered as a group of series or actions that include number 1, food acquisition, number 2, food intake into the oral cavity, and inside the oral cavity we have taste perception and chewing and swallowing to pass the food from the oral cavity to the stomach. So the food acquisition, food intake into the oral cavity, taste perception, chewing by the movement of tongue and teeth. Also, we have the swallowing. And if we look to the feeding behavior, it is affected by a number of factors. And these factors are either internal or external factors. We have internal factor such as the presence of hunger or satiety. If you are hungry, you are going to seek for a food. If you are feeling full, you are not going to seek or to find food. Also, something is going to attract you to the food, which is the taste of the food or the palatability of the food, as well as the health condition. Also, mood is considered as one of the factor or the internal factor that affect our internal need to the food, as well as the atmosphere around the individual. If you are sitting in a restaurant or a cafeteria, what's happening? There is something which is called tendency to eat by the surrounding. Hunger, satiety, appetite, and food reward are the most important factors that regulate our feeding behavior. So the feeding behavior is considered as or affected by hungry, satiety, appetite, and the food reward. Both human and animal engage in a feeding behavior to obtain pleasure, that's known as the food reward, and also to get energy, The pleasure when we ingest food is considered as the food reward, and it is driven by the appetite of food. The other is the obtaining energy from either the carbohydrate or the glucose source of energy or from the fat source of energy, which is the products of the fat hydrolysis or fatty acid oxidation. It is the ketone bodies. It can be used as also as a source of energy. So we are going now to discuss in detail the feeding behavior and the centers affecting it. There are two forms of feeding behavior. The first one dependent on the energy balance inside your body, dependent on the blood level of the nutrients like glucose, like amino acids, and fat oxidation products like ketone bodies. This is known as the homeostasis dependent feeding behavior, and its function is to monitor the energy balance and maintain the energy balance and homeostasis. The second one, controlled by the amygdala and the nucleus accumbens, what are going to do? These are considered as a part of the limbic system, and they are producing something which is called the emotion reward system or the homeostasis independent feeding behavior. Homeostasis independent feeding behavior usually appear when you are sitting in a restaurant or a cafeteria and surrounded by a lot of people who are eating or drinking. And if you are feeling satiety, what's happened after some time? There is something which is called the getting to eat or starting to eat to share them to get the emotion or to increase the reward or increasing the pleasure in your brain, and this is usually affected by the amygdala and the nucleus accumbens. Normally the two feeding behaviors are interacting or acting on the feeding center, which is the lateral hypothalamic nucleus. The lateral hypothalamic nucleus is the feeding center, so both types of behavior are affecting the lateral hypothalamic area. Now, in recent societies or in the modern society, we have surrounded by a lot of information about food coming from the TV, coming from the mobile while you are browsing and looking for something like chefs who are preparing food. What's going? The homeostasis independent control system is usually surpass the homeostasis dependent behavior. So usually we are going to eat when we are not hungry. We are eating by eating snacks or taking like something like the high caloric food or drinks when we are not feeling hungry or thirsty, and this is usually mediated by the homeostasis independent feeding behavior. We have to answer the following questions. Why do we eat? We eat when we are feeling hungry or in the presence of hunger sensation. And hunger sensation is defined as the craving or the urgent need for food or a specific nutrient. And this usually depends on the energy balance. Drop of the nutrients in the blood, like the blood glucose or the presence of hypoglycemia, stimulates the glucoreceptors in the hypothalamus that stimulate the feeding center, and it may be associated with contraction in the stomach known as the hunger pangs, which is sometimes being painful. It is affected also by the eating pattern, how many meals you are eating per day. Also, the temperature and the cold weather can increase the hunger sensation. What do we eat? That is dependent on the appetite, and usually the appetite is a psychological drive that push the person to prefer certain types of food. Why we stop eating? We stop eating in response to satiation, which is the feeling of satisfaction and fullness during a meal. And usually satiation determines how much food is consumed during a meal. On the other hand, satiety is the same feeling of fullness and satisfaction that occurs after a meal, and usually determine how long time passes between meals. Determines how long time passes between meals. The hypothalamus produce homeostasis of the energy balance or maintenance of the energy balance or regulation of the energy balance through two mechanisms. The first one through controlling the activity of the autonomic nervous system and the regulation of food intake. The stimulation of the posterior and the lateral regions of the hypothalamus is associated with increased activity of the sympathetic nervous system, leading to increase in glycogenolysis and lipolysis, and this means energy utilization or burning energy. On the other hand, the anterior region of the hypothalamus is associated with, or its activation leads to increase in the activity of the parasympathetic nervous system, increasing the digestive and absorptive capacity of the gastrointestinal tract energy storage. On the other hand, the hypothalamus has two nuclei that control the feeding behavior. The lateral hypothalamic area is considered as the feeding center or the hunger center that drive us to eat, while the satiety center, located in the ventromedial nucleus of the hypothalamus, stop the feeding behavior or decrease the desire for food. So, the hypothalamus regulates energy balance inside the body through controlling the autonomic nervous system and controlling the food intake. Loading of the body energy reserve. The process of digestion and absorption of the different macronutrients in food leads to increase in the blood level of the monosaccharides, glucose, fructose, and galactose. Glucose will be transported to the liver and skeletal muscle to be used for the glycogenesis. Fructose is metabolized only in the liver, and usually its metabolism leads to increase in the fat synthesis in the liver, and galactose can be converted into glucose into the liver. Amino acid transported by the blood from the intestine to the liver to be used for the synthesis of protein. Fat digestion and absorption in the form of chylomicrons, which is formed by the enterocytes. Chylomicrons transported from the lymph to the blood. The presence of lipoprotein lipase on the adipose tissue cells or adipocytes and skeletal muscle cells uptake the triglycerides from the chylomicrons into the adipocytes and the skeletal muscle to form the chylomicron remnant. Chylomicron remnant go back to the liver, so the liver is going to produce formation of fat or formation of triglycerides, and these triglycerides formed in the liver will be transported in the form of very low density lipoprotein to be transported or to move this to the peripheral circulation of the peripheral tissues. At the same time, beta oxidation of fat occur in the liver, and this is usually associated with increase in ATP production and the formation of ketone bodies. That is the loading of the body energy reserve. Increase muscle glycogen, increase liver glycogen, increase the triglyceride content of both skeletal muscles and adipocytes. In the post-absorptive state, or after the complete digestion and absorption of food, and no food is present in the intestine, the liver starts to produce the glycogenolysis, which is the hydrolysis of the glycogen into glucose, and this glucose will be usually used as a source of energy for the brain. Also, the fat present in the liver, or the fatty acid oxidized into the liver, leads to the formation of ATP, and some of the acetyl-CoA coming from the fat or from the fatty acid oxidation will be converted into ketone bodies, and these ketone bodies can be used as a source of energy also for the brain and to the heart. The adipocytes, or the adipose tissue, usually in the response to the hormone sensitive lipoprotein lipase, mainly to the glucagon, which increase during the process of fasting or in the post absorptive state, release triglycerides to the blood, and these triglycerides will be transported to the liver to be used as a source of energy and also for the formation of ketone bodies, or transported to the skeletal muscle to be used as a source of energy in the absence or with a decrease in the blood glucose. So that is the emptying of the body energy reserve. Emptying of the body energy reserve. Now we are going back to the hypothalamus and looking for the feeding center and satiety center in the hypothalamus. The lateral hypothalamic nucleus in the hypothalamus is considered as the feeding center, while the ventromedial nucleus of the hypothalamus is considered as the satiety center. So we have the dual center model of feeding behavior. The dual center model of feeding behavior, this means that the sensation of hunger is dependent on the interaction between two types of nuclei in the hypothalamus. If we have lesion in the lateral hypothalamus, or in the feeding center, the animal or the rat is showing hypophagia. Bilateral destruction of the lateral hypothalamic nucleus is associated with hypophagia and loss of weight. On the other hand, if we have bilateral damage of the ventromedial nucleus of the hypothalamus or loss of the satiety center, this is associated with hyperphagia and increase in the body weight. So the feeding behavior or the sensation of hungry or satiety is dependent on the interaction between two types of neurons in the hypothalamus. The neurons located in the lateral hypothalamus, and that is including or known as the feeding center, or the nucleus or the ventromedial nucleus of the hypothalamus, which is considered as the satiety center. Other hypothalamic nuclei controlling the feeding behavior: bilateral damage of the paraventricular nuclei associated with excessive eating. The arcuate nucleus is found to be the site of action of the gastrointestinal tract hormones and adipose tissue hormones like leptin, and the gastrointestinal tract hormones like cholecystokinin. This means that the information coming from the adipose tissue and from the gastrointestinal tract converge on the arcuate nucleus to regulate the food intake and feeding behavior. So this means that the arcuate nucleus, paraventricular nucleus, and the lateral hypothalamic nuclei, ventromedial nuclei, are interacting together to control the feeding behavior. In the present slide, we are going to study the lipostatic theory, the glucostatic theory, and the aminostatic theory. We have three theories regarding to the level of the nutrients in the blood. The first one is dependent on the blood glucose level, second one on amino acid, and the third one, which is the lipostatic, is dependent on the metabolites of fat oxidation, mainly the ketone bodies. Increase the plasma level of these nutrients is associated with the production of satiety and the production of the anoxygenic neurotransmitters in the brain, in the hypothalamus. So usually this will be associated with stoppage of feeding and inhibition of food intake or inhibition of feeding with the sensation of satiety with stimulation of the sympathetic nervous system. The sympathetic nervous system at this point is going to stimulate the process of lipolysis and glycogenolysis to produce the breakdown of energy. So in this condition, the body is going to produce satiety with stimulation of the sympathetic nervous system. At the same time, the adipose tissue start to release leptin, and leptin in this condition is going to the hypothalamic satiety center located in the ventromedial nucleus, affecting the arcuate nucleus through its receptors, which is located on the arcuate receptor, to produce more satiety. In this condition, we can find that with the increase in leptin, there is an increase in satiety. And this condition was explained in persons who go into prolonged fasting. They can go into prolonged fasting for 24 hours, 36 hours, 48 hours without any feeling of hungry, and this can be explained by the increase in the level of ketone bodies coming from the process of lipolysis, with increase the production of leptin that will produce the satiety sensation. On the other hand, if we go to the condition, if we have a decrease in blood glucose mainly, or hypoglycemia, decrease in the circulating level of the amino acids, usually we have a stimulation of certain types of neurotransmitters, or increase in a specific type of neurotransmitter called the orexigenic neurotransmitters, and this usually associated with the sensation of hungry, and the body is going to shift with the storage of energy, and this usually associated with the stimulation of the parasympathetic nervous system that increase the digestion and absorption of food from the gastrointestinal tract. Also, it stimulates the pancreas to release insulin, and insulin is known as a fat storage hormone, which is going to increase the entry of glucose inside the cell through the insertion of the GLUT4 to the cell membrane of the cell, so increasing the energy storage. Here we have a connection between, or studied the connection between, the regulation of food intake and the interaction of the sympathetic and parasympathetic nervous system in response to the changes of the blood nutrients, in response to the change of the blood nutrients. After we studied the mechanism, or the glucostatic theory, aminostatic theory, and the lipostatic theory in the regulation of hunger and satiety by affecting the orexigenic and anorexigenic neurons in the hypothalamus, now we are going to another regulation, which is the regulation of the quantity of food intake. Regulation of the quantity of food intake, or the amount we are going to eat, is determined by short-term regulation, and the short-term regulation usually determine the meal, or the size of the meal, while the second type of mechanism of regulation, or the long-term regulation, that usually monitor the energy balance and regulate or control the body weight. So we have two types of regulation: short-term regulation that determine the size of the meal to prevent overeating, and the second one is the long-term regulation that's going to monitor the energy balance and control the body weight. So let's go and discuss in details. Short-term regulation of food intake. The short-term regulation tends to control the size of the meal to prevent overeating, mediated by two pathways. The first one is the gastrointestinal distension. Distension of the stomach and the duodenum send inhibitory impulses through the vagus nerve to the feeding center to inhibit its activity and stop feeding. The second mechanism is dependent on hormones. Hormones, or gastrointestinal hormones, are classified into two categories of hormone. The first one is the hormones that suppress feeding, including cholecystokinin, peptide YY, and incretin. These hormones are coming from the small intestine in order to suppress feeding. Peptide YY mainly secreted in response to amino acids and protein in the intestine. Fat stimulates the secretion of cholecystokinin, and incretin appear or increase in response to presence of glucose in the small intestine. The second category of hormones, or the second class of hormones, is the gastrointestinal hormone that increase feeding, and this include only one hormone which is coming from the stomach, which is the ghrelin hormone. Ghrelin coming from the stomach is one of the hormones that increase the feeding, is increasing the feeding. The gastrointestinal hormones that suppress feeding include the fat signal or cholecystokinin that is secreted from the cells of the small intestine or the enteroendocrine cells, which is mainly known as eye cells in response to fat or fat digested products. Cholecystokinin inhibit the feeding center through the melanocortin pathway or through activation of the melanocortin pathway. Second hormone is the protein signal or peptide YY, which is secreted from the gastrointestinal tract cells, but mainly from the cells of the ileum and colon. Usually, this peptide YY reach to its peak or increase to its maximum level 1 to 2 hours after ingestion of a meal. High level of peptide YY was observed with the high protein content or meals that contains high protein content. So high protein intake increase the peptide YY and this hormone suppress feeding. The third hormone, which appear in response to the presence of carbohydrate in the small intestine, is known as the glucagon-like peptide. This glucagon-like peptide is belonging to a group of hormones known as incretin. This glucagon-like peptide stimulates the pancreas to release insulin. The increase in insulin usually suppress the postprandial hyperglycemia or the increase in the blood glucose after meals. Also, this glucagon-like peptide is also suppress feeding. Another drugs or newly there is a group of drugs working through the increase in the incretin or mimicking the effect of the glucagon-like peptide used for the treatment of type 2 diabetics and also for the obese people by the inhibition of the feeding center and increasing the satiety. So the gastrointestinal tract hormones that suppress feeding include the fat signal is cholecystokinin, peptide YY, and incretin. And it is noticed that the meals which is rich in fat and protein are usually prolonged or increasing the satiety for long period. So the duration between meals can be prolonged by increasing the fat and protein content in diet. The hormone which increase the process of feeding or increase the food intake is the ghrelin hormone. Ghrelin hormone increase in between meals and reach to its peak just before the meal. After food intake, it go and drop to its lower level or to its lowest level, then it gradually increase in between meals. It is secreted from the exanthic cells of the stomach and to less extent from the intestine. The injection of the ghrelin hormone into experimental animals increase the feeding behavior or increase their food intake. This means that it is considered as an erexogenic hormone. Ghrelin also appear to suppress fat utilization in the adipose tissue. Ghrelin also appear to suppress fat utilization in the adipose tissue. So usually the persons who are going to make a surgical operation to reduce the size of the stomach or the bariatric surgery, this usually produce a wonderful effect on their body weight and decrease their appetite. Why? Because the removal of large part of the stomach removes the stimulus or removes the ghrelin hormone. So the people with resection of the stomach or gastrectomy, what's going to them after that? Drop in ghrelin, so they have a marked drop in appetite and a marked loss in body fat or body weight. So ghrelin increase the appetite, ghrelin drop in the appetite, while the satiety hormones increase in cholecystokinin, glucagon-like peptide, peptide YY. All of them when increase they induce the satiety, when they drop they decrease or there is a decrease in satiety, increase in the appetite. So ghrelin increase is associated with the decrease in cholecystokinin, glucagon-like peptide, and peptide YY, and this will increase the appetite and increase the feeding behavior. Oral receptors meter the food intake. When an animal with esophageal fistula produce chewing of food and swallowing without passage of the food to the stomach for a specific time, what's going the animal is going to show satiety or have stoppage of the process of feeding. This means that the stimulation of certain types of receptors in the oral cavity and these receptors are going to send impulses to inhibit the feeding center. This means that there is an inhibition of the feeding center in the hypothalamus coming from the oral cavity receptors. So after a specific time of chewing food, if you are going to prolong the chewing time, what's going this share in the induction of satiety by inhibition of the feeding center. So usually they are going to advise the people with obesity to produce more chewing of food in their oral cavity to give a specific time for the oral receptors and the taste receptors in the tongue to send impulses to the lateral hypothalamic area and inhibiting it, and this will share in the induction of satiety. This mechanism is not prolonged. It gives a shorter duration of inhibition than the inhibition caused by the gastrointestinal filling. So the oral receptors can share in the short-term regulation of the feeding behavior by inhibiting of the lateral hypothalamic nucleus in the hypothalamus. Now we are moving to the long-term regulation of food intake. The long-term regulation of food intake is dependent on the signal coming from the adipose tissue to affect the hypothalamus in order to regulate the fat storage or the energy storage and body weight. The adipocytes are known or previously known as a site of fat storage and energy storage, but they have also the capacity to secrete a certain types of chemical factors known as adipokines or the hormones coming from the adipose tissue. One of these hormone is the hormone leptin. Leptin hormone released from the adipocytes, and usually the action of leptin is to cross the blood-brain barrier and going to bind with its specific receptors located in the hypothalamus, especially in the pro-opiomelanocortin receptors of the arcuate nucleus and on the paraventricular nucleus and the pro-opiomelanocortin neurons in the arcuate nucleus. When leptin crosses the blood-brain barrier and is going to bind with its specific receptors in the arcuate nuclei, is going to produce suppression of food intake and producing satiety. Defect in leptin, inability of the leptin to cross the blood-brain barrier, absence of leptin receptors, this will lead to weight gain. Leads to weight gain. So now we are going to look for the action of leptin. The action of leptin. The action of leptin hormone is mainly on the hypothalamus and pancreas. On the pancreas is going to decrease insulin secretion by the pancreatic beta cells. It will decrease insulin secretion by the pancreatic beta cells. At the same time, on the hypothalamus is going to decrease the production of the orexogenic neurotransmitters or the neurotransmitters that increase the food intake, mainly neuropeptide Y and agouti related peptide. Neuropeptide Y and agouti related peptide. On the other hand, is going to activate the pro-opiomelanocortin neurons, causing the release of alpha melanocyte stimulating hormone and activation of melanocortin receptors, and this usually produce the satiety or the sensation of satiety. Increase production of substance such as the corticotropin releasing hormone that decrease food intake. Increase the production of corticotropin releasing hormone, decrease food intake. Also, it increases the activity of the sympathetic nervous system, and this will increase thermogenesis and increase the basal metabolic rate. So leptin decrease the production of the orexogenic neurotransmitter, neuropeptide Y and agouti related peptide. Activation of the pro-opiomelanocortin pathway, activation or increase in the alpha melanocyte stimulating hormone and activating melanocortin receptors. Increase in the formation of corticotropin releasing hormone that decrease food intake. Also, it produce increase in the sympathetic nervous system activity, and this will lead to increase in the thermogenesis and metabolic rate. Now we are moving to the lipostatic hypothesis for regulation of eating and body weight. The lipostatic hypothesis for regulation of eating and body weight. With increase in the body fat, there is increase in the circulating blood level of leptin. Increase the fat accumulation in the body. This will be associated with increase in the circulating level of leptin. And leptin cross the blood-brain barrier to bind with specific receptors on the arcuate nucleus. Binding of leptin to its specific receptors on the arcuate nucleus produce increase in the catabolic neurotransmitters, mainly alpha melanocyte stimulating hormone and cocaine amphetamine regulated transcript. So there is increase in the MSH, which is a melanocyte stimulating hormone, and CART, which is cocaine and amphetamine regulated transcript. These neurotransmitters are going to bind to the receptors located on the lateral hypothalamic nucleus and the paraventricular nucleus. On the lateral hypothalamic nucleus, through the melanocortin receptors subtype 4, is going to produce an inhibitory effect on the lateral hypothalamic nucleus. Inhibitory effect on the lateral hypothalamic nucleus, and this will lead to decrease in appetite and food intake. The catabolic neurotransmitters affecting the melanocortin 4 receptors, and this will depress the food intake through inhibitory effect on the lateral hypothalamic nucleus. On the other hand, they have a stimulatory effect on the paraventricular nucleus of the hypothalamus. They are going to stimulate the paraventricular nucleus of the hypothalamus to increase the secretion of corticotropin releasing hormone, thyrotropin releasing hormone, and oxytocin. This humoral response increases the metabolism and energy expenditure. So there is increase in the corticotropin releasing hormone, thyrotropin releasing hormone, and oxytocin, and their effect is to increase the metabolic rate and the energy expenditure. At the same time, the stimulation of the paraventricular nucleus increases the activity of the sympathetic division of the autonomic nervous system, leading to increase in thermogenesis. So usually leptin is going to produce inhibition of the food intake and appetite with increase in the thermogenesis. So again, with increase in the body fat or increase in the circulating leptin level crossing the blood-brain barrier, affecting the arcuate nucleus. The arcuate nucleus, when affected by leptin, there is increase in the catabolic neurotransmitters, melanocyte stimulating hormone, and the cocaine and amphetamine regulated transcript to inhibit the lateral hypothalamic nucleus and stimulate the paraventricular nucleus. The discovery of leptin brings us to a question: how can leptin reduce body weight or induce the weight loss? The discovery of leptin is associated with the discovery of the knocked out mice, which is known as op mice. It is a mice or a rat with a defect in the generation of leptin. These rats cannot form leptin. Another types of rats with a defect in leptin receptors in the hypothalamus. Both types of rat have severe obesity and increase in body weight. With restoration or injection of leptin directly into the brain through the intracerebroventricular injection into the hypothalamus directly, produce fat loss and allow the rat to become again into a normal body weight. So leptin is known as the hormone that decrease the body weight. And if we look to the people with obesity or morbid obesity and increase in the body weight, they have excess adipocytes, and consequently there is increase in the leptin in their blood. But what is the problem? The problem is leptin resistance. So the people with excess adipocytes, excess leptin, they are going to eat more and more. There is no inhibition of the feeding behavior, and this lost inhibition of the feeding behavior continue, and they are going to continue to have something like disorders in the eating behavior. Usually the main cause of this leptin resistance is hyperinsulinemia. When the people are eating sugary foods or excess carbohydrates or excess intake of food, there is increase in the insulin production in the blood. Increased insulin is usually associated with inhibition of the leptin action. It prevents the leptin from producing its action on the arcuate nucleus. So absence of binding of leptin on the arcuate receptors caused by the hyperinsulinemia. So the main treatment to reduce the body weight of these obese people is to reduce the insulin level. If you decrease the insulin level, the leptin will start to work again, so it will inhibit the feeding, and this usually is associated with weight loss. So the main treatment of the obese people is to reduce the insulin level. Decrease the body fat, decrease the plasma or decreasing the circulating plasma level of leptin, Or there is leptin but cannot cross the blood-brain barrier. Leptin cross the blood-brain barrier but couldn't bind to the receptors on the arcuate nucleus. The binding occurs but there is a defect in the pathway with increase in the ghrelin level coming from the stomach. This will induce the formation of anabolic neurotransmitters like neuropeptide Y and agouti related peptide. These anabolic neurotransmitters go to the lateral hypothalamic nucleus to produce a stimulatory effect to increase the orexin and melanin concentrating hormone stimulating the food intake. So increase in the ghrelin with a defect in the action of leptin will increase the formation of neuropeptide Y and agouti related peptide to produce a stimulatory effect on the lateral hypothalamic nucleus to increase the food intake through the melanin concentrating hormone and orexin. At the same time, they are going to produce an inhibitory effect on the paraventricular nucleus, producing inhibitory effect on the paraventricular nucleus, and this will lead to activation of the parasympathetic nervous system, to decrease energy expenditure and increase energy storage, to decrease energy expenditures and increase energy storage. Also, they are going to increase the food intake through increasing the activity of the parasympathetic nervous system on the gastrointestinal tract. So this means that leptin is going to induce satiety, ghrelin is going to induce food intake through a change in the neurotransmitters at the level of the arcuate nucleus, through activating of either catabolic neurotransmitters by the effect of leptin or anabolic neurotransmitters with the decrease in leptin and increase in ghrelin. Role of temperature in regulation of food intake. There is a connection between the heat regulating center in the hypothalamus and the arcuate nucleus in the hypothalamus, or the feeding center in the hypothalamus. With exposure to cold, there is increase in the food intake. Usually, if we perform exercise in cold or the presence of cold weather, this will produce inhibition to the leptin of action and inhibition of the anorexogenic hormones coming from the gastrointestinal tract to increase the food intake. This will help to increase the metabolic rate. As we know, the increase in food intake increase in the thermogenesis. Also, this will provide fat for insulation. Increase food intake, increase the accumulation of fat in the adipose tissue, and as we know, the fat act as insulator to decrease the heat loss. So usually in exposure to cold there is increase in the food intake, and with exposure to hot there is decrease in the energy intake. That is dependent on the connection between the temperature regulating center and the feeding centers located in the hypothalamus. Feeding and eating disorders. Binge eating disorder. Frequent episodes of consuming very large amount of food but without behavior to prevent weight gain, such as self-induced vomiting, and that is the same like the bulimia nervosa but with behaviors to prevent weight gain, such as self-induced vomiting. So the patient or the subject with binge eating disorders is going to consume large quantities of food without control of body weight and don't look to the body weight, so they usually not producing the self-induced vomiting. On the other hand, the bulimia nervosa is going to produce this to control their body weight. Princess Diana was complaining from the bulimia nervosa. She eats too much and after she finished she go to produce the self-induced vomiting. Another type of feeding disorder is the anorexia nervosa. Those people who are monitoring their weight and they are afraid from getting overweight and obese, they have something like obsession with weight gain and they frequently stop eating and leading to marked loss of the body weight. This condition is known as the anorexia nervosa, characterized by the marked loss of the body weight. Their body mass index may be less than 17.5%. So that is the feeding and eating disorders. At the end, thank you.